Okay, so welcome back to all of you. So we're going to continue on with our lectures here. Okay. And again, I know some of you don't really love the lecture style, but I'm trying to prepare you for university. And if you're going to science, you're going to get lecture style. And you're not only going to get these little mini lectures that I'm giving, which are 20 at most 20 to 30 minutes. You're going to be getting sometimes 3 hours of lectures, which is very, very difficult. You have to sit there and listen to some guy or girl talk that may not be the most interesting, and you have to try and pay attention for 3 hours. So although I'm not, talking for 3 hours, thank God, I'm trying to prepare you for this new reality that you're going to have to face next year. So it's very good practice. So I highly suggest you print out the slideshow. If you don't want to print them out, you can use your iPad and go through it. I just prefer myself to print them out. And when you do print them out, don't print them out one slide per page. That will take forever. You'll have a book every time you go. But print them out like 6 slides per page or 3 slides per page. That's normally what my students do. Okay? All right, so what we're going to do today is we're going to talk about basically how nerves work. So we've already gone through the general way that homeostasis works, the 2 feedback loops. Then we went through the general organization of the nervous system. Okay, we kind of just went generally. We're still going generally. We're going to talk about generally how nerves work. So the nervous system works on neurons, which are the cells of the nervous system. And the cells look really weird. They don't look like normal cells. They kind of have like these little dendrites. It's kind of like a star with like a long tail on the end that we call an axon. Right? We're going to talk about how those cells actually communicate to each other. Like when we want to send an impulse, like from my brain and into my hand, how does that travel along? Well, it's going to travel along a neuron. Okay? And a whole bunch of neurons together are considered a nerve. So it's really going to travel down a nerve from my motor cortex all the way down in my spinal cord, which is still CNS. Okay? Brain is spinal cord, CNS. And then it'll go out my peripheral nervous system and down my arm, and then basically to my effector and move my hand. Okay? So today we're going to talk about basically how those nerves communicate. Now there's 2 main parts with nerve communication. There's action potentials, and then there's synaptic communication. I'm going to focus on synaptic communication. I'm not going to ask you really anything on action potentials. Okay? If you take exercise science, you'll learn a lot about them. You'll also learn a lot about them in second year. Okay? The idea of an action potential is really just using a bunch of channels, sodium and potassium channels, to create a difference in charges from positive and negative on the inside and outside of the cell. Okay? But I'm not going to ask you a lot about those. What I'm going to really focus on is synaptic transmission. Okay. So when you look at my slideshow, I am going to have some stuff on action potential, but you can just skip right over it because I'm not going to ask you about it. So how do nerves work? Okay. So nerves are specialized cells. How do they perform their function? Like sending signals from the peripheral nervous system to the central nervous system, to compute what exactly is happening, and then back to the peripheral nervous system. If we go back to our feedback loop, okay, and again, all homeostasis works on feedback loops, we would have the sensor, the controller, and then the effector. Okay. All right, so we presently know how the nervous system is organized in the different major parts, the CNS, the PNS, the somatic PNS, the autonomic PNS, etc. We know the basic parts, the neurons and the glial cells. Okay. And we know the different types of neurons based on their structure and function. Again, I'm going to be focusing on function, not structure. The next step is going to be how neurons work. So the electrical nature of nerves was first realized when, okay, we're just going to skip over this. So these are action potentials. Okay, here we go. So this is what we're going to be talking about. It's basically when a nerve terminates and the next nerve starts. Okay? So here is our neurocell here. Okay, so here's a neuron. You can see this whole entire thing is a cell. From this part right here, the axon terminal, all the way down the axon and then up to the cell body. Okay? This is an entire cell. Now, a couple things important to realize is that we function on electricity. Okay? It's sometimes weird to realize that we're humans, but we actually function on electrical impulses. So how neurons communicate to each other is they send basically an electrical impulse from here all the way down their body and then to the end on the axon terminal. Okay? Then something weird happens, okay? Because a lot of neurons don't connect from, say, your brain. This isn't your brain and this is your muscle. There's going to be a break where one neuron ends and then the next one starts, okay? So these little breaks are called a synaptic cleft or a synapse. And it's a very interesting area because what's going to happen is we're going to have an electrical message. Okay? So when I'm inside my neuron, this is an electrical message traveling down here. So it would be like, oh, I want to move my arm. So that message gets sent through electricity, just like how The internet works sending messages through electricity, or a phone works sending messages through electricity. So I'm going to get a message, okay, and I'm going to send it through electricity through my neuron. Okay. Well, when my neuron breaks, okay, and then before the next neuron starts, there's a little space in between here. They don't connect completely. And in this little space, I'm going to transfer the electrical message into a chemical message. And that chemical message is going to be in the form of a neurotransmitter. So I'm going to have a chemical message that just sends that. So the electrical message turns into a chemical message. The chemical message is able to cross this little gap, and then it turns back, in most cases, back into an electrical message, and then goes down, and then may go to a chemical, and then back to electrical. Okay? So the types of messages that our nervous system sends, and again, those are kind of immediate messages, can be electrical, and then they go into chemical, and then they most of the time go back into electrical. Okay? So the nervous system is responsible for communicating direct messages. The nervous system sends a message via electrical signals called action potentials. So an action potential is going to travel along here. Action potentials carry electrical messages down the nerve fibers where they are transferred to a chemical message, which would be a neurotransmitter. The message is then transferred back to an electrical message in most cases on the next neuron. Okay. Now, when we send a signal down a neuron, it's very important that we don't lose that signal. So if I send a message here that I want to move my arm, I want to make sure that that message gets all the way down to my axon terminal. If it has to go through a neurotransmitter, then to a next axon. But we want to make sure the message isn't lost halfway through here. So some nerves have their axons coated in material called myelin. So this little yellow material is considered myelin. And the whole purpose of myelin is basically an insulator. Okay? It's responsible for ensuring that the message gets from, say, the cell body all the way to the axon terminal and isn't lost in translation at all. Okay? It's basically an insulator of an electrical signal. Okay? So myelin serves two main purposes. It ensures the signal is not lost during travels, okay, so that you don't lose a signal here and then you never get that signal to your arm to move, or increases the speed at which the signal can be sent. Okay? Now, just before I move on to there, okay, if you've ever heard of MS or multiple sclerosis, or people who have MS, they've lost their ability to have this myelin. Okay, so they're losing some of their myelin. And when they lose some of their myelin, what happens is, in an MS patient, you may inside your head say, I want to move my arm. Well, you go to try and send the action potential to move your arm down your neuron, but it never gets to the end. Because you don't have myelin, the signal comes off here. So a person who has MS may think, I want to move my arm, and they may start firing neurons in order to move their arm, but because the action potential gets lost early on, it never gets down to their hand or to their arm, and they can't actually move their arm. Okay? So that's just an example of a disease caused by lack of myelin. Okay, so we're going to focus right on this, nerve-to-nerve transmission. So oftentimes a single nerve fiber does not run the length of the body, i.e. a single nerve from your brain all the way to your arm muscle. Oftentimes one nerve will excite another nerve, or a group of nerves. Okay, so this next series of slides will explain how one nerve communicates to another nerve, a process called synaptic transmission. Okay? Now this little website right here, I highly suggest you take a look at it. Okay, it's going to go through a synaptic transmission very, very slowly. Okay, it's really, really, really good. Just make sure when you go to it, you click on synaptic transmission. Okay, but really, really good website. Highly suggest it. Okay, so nerves need to communicate. So nerves need to send information to one of these three spots. Okay, so when you send an action potential, and here's our nerve, when it comes down and it lands here at the axon terminal, it can now go on to three different areas. It can go on to another nerve. Okay, and then that's just going to be via synapse, it's going to go to another nerve, and then action potential. Okay? It can go to an effector, which would be like a gland, an artery, a vein. Okay? So then it can start contracting an artery, contracting a vein. Okay? And that would be it could kind of look similar to this, where say here's an artery, the message comes down, you got to constrict that artery, the neurotransmitter will release, okay? That neurotransmitter will basically attach to the artery and then cause it to constrict. Okay? And the last one is a neuromuscular junction, and that's when it ends on a muscle. So here's an example of a neuron coming down. It's embedded in the muscle. Okay, so that action potential will come down, get converted to a neurotransmitter. That neurotransmitter will come in and basically cause the muscle to contract. So an action potential developed in the cell body travels down the axon away from the cell body until it reaches the synaptic terminal. And that's kind of like our synaptic terminal right at the end here. So here we've got the electricity kind of shown by a lightning bolt. It's coming down, and we're ending here. And it looks like on here we have a gland or an effector, a muscle. We don't have another nerve that's going to be sending another signal. Okay. At the synaptic terminal, one neuron doesn't touch another neuron or a muscle or an effector. There's a small space between the end of the neuron, okay, and the start of the nerve muscle. So this little space right here. Okay. We call this little space the synaptic cleft. So the synaptic cleft is too large for an electrical signal to just jump. Okay. So the action potential can't just jump across this little space. Therefore, the electrical signal, the action potential, must turn temporarily into a chemical signal, which is a neurotransmitter, to cross the synaptic cleft. So the action potential is going to cause the release of a neurotransmitter which comes across that synaptic cleft, and there's receptors on the other side that receive that neurotransmitter and either carry on the message or perform the effector. So here's a really, really good picture of everything that's happening. Okay, so this is a lot of detail. I would not necessarily expect you to know all of this detail, but the basics I would. So you have the nerve impulse coming down in action potential. Okay, when that action potential comes down, okay, it causes calcium channels to open up. Okay, I'm not going to ask you about the calcium channels, but just so you know a thorough understanding of it, causes the calcium channels to open up. Calcium's going to rush in. Okay, when calcium rushes in, it causes the neurotransmitters to start basically moving towards the synaptic cleft. So these calcium ions cause these neurotransmitters, but through the use of bonding and forces, to move to the synaptic cleft. And once they get there, the vesicles release. This is kind of exocytosis. If you remember exocytosis from our first unit, where you have a vesicle, okay, of a chemical, most likely a fluid, okay, and you're going to release that vesicle outside the cell, okay, through exocytosis. Okay. So that neurotransmitter then goes across through the synaptic cleft, and it'll join onto all of these receptors on the other side. Okay. Now these receptors could convert it back to an action potential if it's another neuron here, or it could cause a muscle contraction if this was a muscle, or if it was an artery or a gland, it could cause a reaction. Okay. Now once this neurotransmitter has basically responded to these receptors, the last step is these neurotransmitters have to disconnect from the receptors and then go back inside this terminal. So what's going to happen is this little neurotransmitter, which could be dopamine, it could be acetylcholine, it could be serotonin, it's going to release from this and then go back inside a vesicle and then back into the terminal here. Okay? So the neurotransmitter is going to release, go inside the synaptic cleft, and then go back in. And then the next action potential comes down, it's going to release its neurotransmitter in here, and then reabsorbed back by this thing. Okay? Now something like a drug like cocaine, okay? How cocaine works is, you know how I was just talking about how that neurotransmitter is going to release and then have to be reabsorbed? Well, cocaine prevents the reabsorption of most likely dopamine, okay, a feel-good hormone. So what happens is that neurotransmitter is stuck in the synaptic cleft, so keeps firing good feelings, okay? So it's basically constantly telling you that you have a good feeling. Normally, if someone says, oh, you look really nice today, right? Someone says, oh, normally you look really, really nice today. What's going to happen is you're going to get an action potential that says, oh, they said I look nice today. It's going to come down your brain. It's going to release a little bit of dopamine. Dopamine's going to come across here, and it's going to make you feel good, okay? Dopamine's going to be reabsorbed, and that feeling's going to go away. When someone tells you to look nice, you don't feel good for 10 years. You only feel good for an instant because that dopamine released and then got reabsorbed. Well, if you take cocaine, and don't take cocaine, but if you do take cocaine, okay, what's going to happen is that you're going to release that neurotransmitter, and it's going to continuously make you feel good. Okay? Now, this is a huge problem. You may think, oh, well, it makes us feel good. What's the problem with cocaine? The problem is you start running out of neurotransmitter. If you constantly have it inside the synaptic cleft, it starts to burn out, and then you lose some neurotransmitter. So if you continuously do cocaine, over time, you're not going to be able to get happy over things. Because normally what's going to happen is someone says, nice, you're going to release that neurotransmitter, dopamine, but you're not going to have any dopamine left because you've been using cocaine so much. Okay? So definitely do not be using any illicit drugs. And that's one of the problems with it, is the elimination of neurotransmitters in our brain. Okay? All right. So neurotransmitters, sorry, just kind of finishing up here. Neurotransmitters have the ability to excite the postsynaptic membrane, open NA channels, or inhibit it. Okay? An activity is, or an example is acetylcholine is a neurotransmitter between the nerve and a muscle fiber. Okay? Tylenol is an example of a drug that tries to inhibit the neurotransmitter acetylcholine. Okay? So there's some popular neurotransmitters. This is from my in class where they look on page 360. I'd like you to look up some neurotransmitters just to know what some of them are. Okay, I'm not going to ask you much more detail than I explained there, knowing that when nerves fire, they go from electrical into a chemical as they cross the synaptic cleft. Right, and that's going to be the big thing that I'd like you to know for today. There's another good video down here on how cocaine works. It's very interesting to watch. I highly suggest you watch that as well. But that's it for nerve-to-nerve communication. Okay. And the big thing there is when we talk about nerve-to-nerve communication, I won't be focusing on action potentials. I'll be talking about that synaptic cleft. Okay, so make sure you go through that, and if you have any questions, make sure you ask me. Again, watch those videos in there. Okay, this isn't a very long video itself, so make sure you actually go in and watch those YouTube videos. And if you don't want to type in those addresses, then just go to YouTube and type in synaptic transmission or synaptic cleft transmission or synapse to synapse. Okay, and make sure you watch some visuals on